peptide bonds join amino acids together. It's just a fancy word for a polar covalent bond. The only reason we have the word peptide bonds is because we're dealing with peptide proteins. Any other place, it's the same bond, it just has a different name because it's not protein.
So the peptide bonds are gotten with amino acids or molecules?
No. Some of them have, this is kind of weird. Some of them have non-polar ends that want to be away from water. Some of them have polar ends that want to be near water. So you get weird molecule shapes because parts of the molecule want to be close to other parts of the molecule, but certain parts want to be far away from the other part of the molecule. That's what helps give proteins their shape. If you learn nothing else about biology in this class, learn that shape is everything. All of these proteins are shaped to let them do their job. You heard of form follows function? Something is shaped to let it do its job. Proteins are the perfect example of that. The analogy they use all the time is lock and key. You know, one lock, one key, boom, fits. That's like this. Proteins work because of their shape. All these chemical reactions we're going to talk about work because of the shape of the proteins that are catalyzing. Ooh, what's catalyze me?
Take a break. No?
You're on the right track. You're on the right track. When you catalyze the reaction, go ahead, either of you. Yeah, beautiful, increases reaction rate, makes it go faster, makes it more likely to happen. Any kind of reaction, you add an enzyme, add a catalyst, and it'll go faster. Sometimes it will happen versus it won't, other times it'll happen faster. So, we'll see in a minute. Serving as a catalyst is a major function of proteins. But first of all, we gotta talk about how they're put together.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical.
Wait, what's this? What is hemoglobin? What does that do? Yeah, it carries oxygen. Where do you find it? Inside red blood cells. Red blood cells are just bags of hemoglobin. That's all they are. There's no nucleus or anything. And they have hemoglobin on the inside. Each hemoglobin molecule can carry 4 oxygen molecules. Fun fact.
Reactions happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom.
Okay, the carboxyl group and the amino group, everybody has that. Every single amino acid has a carboxyl group, that's what makes it an acid. And an amino group, that's what we call an amino. And the side chain here, this is where they're different. This is where they're different.
The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar amino acids interact well with water.
So we got the hydrophobic dudes, we got the hydrophilic dudes, and they want to stay far away from each other, and they want to be close to their buddies.
Charged amino acids interact with oppositely charged amino acids or with other molecules. Primer.
Look in your notes. 4 structural levels of organization, protein shape. Primary structure, secondary structure, tertiary structure, and quaternary structure. That's 4 different levels of structures. I bet there's a test question or 2 about this, just saying.
Primary structure. primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide.
So what reaction joins these guys together? Yeah, dehydration synthesis. That's how you put these guys together. Now this is all fancy, it's got a whole bunch of stuff going all different directions. So let me simplify this a little bit. That's an amino acid. Doesn't matter which one. That's another amino acid. Another amino acid, another amino acid. That's what they mean when they say primary structure. Now there's any one of 20 or 21 different names. The order that they're put together defines their shape. Because the hydrophobic ones want to be near the other hydrophobic ones, and the hydrophilic ones want to be near the other hydrophilic ones. The positive charged dudes want to be near the negative charged dudes. And everybody wants to stay away from its evil twin, so to speak. So the order you put these guys in, that's primary structure. And that defines the shape it's going to be. Does that make more sense than that? You guys okay? Is everybody cool with primary structure? Just the order of the amino acids. Now they're nicely pointing out that DNA determines the order of amino acids. You guys all heard DNA is like the master key, right? It's like the thing that does everything. Well, all DNA really is recipe book. DNA is just a recipe book. How many guys keep, or your mom keeps, or more likely your grandma keeps, this metal box with 3x5 cards with a bunch of recipes on it? You know what I'm talking about? Or how many guys have a cookbook at home? How many have anything by Alton Brown? Alton Brown rocks. If you don't know about Alton Brown, you should watch. If Alton Brown gets into his recipes, best brownies you'll ever make, promise. You don't even need to put anything on it, they're good all by themselves. But DNA is just a collection of protein recipes. That is the recipe for protein. Primary structure is the recipe for protein. And Anthony Bourdain fans got it. He's freaking awesome. I love him. All right, primary structure. Order of amino acids.
On its form. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids.
See, it looks kind of like a spring. You get these dudes lined up like this. You'll have amino acid, 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 boom, boom. And they line up kind of like a spring. And we say they form an alpha helix. That's one example of secondary structure. The other example is called a beta pleated sheet, or a beta sheet.
Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids.
So we got our beta sheet, kind of like this. That's the secondary structure. This happens, like it says up there, because these dudes like to be near each other. To get these dudes to want to be near each other, you have to have them in the right order. Primary structure causes secondary structure. Because some dudes wanna hang out with other dudes, some dudes wanna be far away. That make sense to everybody?
Tertiary structure of a protein is the three-dimensional shape of the protein.
See here we got an alpha helix, couple of beta sheets, and that makes a single strand protein.
The shape is determined by characteristics of the chain. Many proteins form globular shapes with hydrophobic side chains sheltered up inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane.
Remember these dudes? What are these called? Phospholipids. Yeah, remember they have the head and the two tails? All the heads are near the water. All the tails are near each other because that's hydrophobic in there. And all these heads are near the water too. This sticks in here because this is also hydrophobic. It wants to be with its buddies, with its hydrophobic buddies. Does that make sense to everybody? All right, so we have a protein, a single protein chain. Let's give it an alpha helix, and a beta sheet, and another alpha helix, and another sheet. You kind of do this. This is tertiary structure. One chain glommed up. That's tertiary structure.
Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes.
Okay, let's add another one. Quaternary structure is when you have two or more protein chains together. So here we got that protein, and we also have that protein. I have two different protein chains there. That's quaternary structure. Four, 3, 2, 1. Primary, secondary, tertiary, quaternary structure. How do you feel about that? Everybody okay? Do you have any questions? Now's a good time to ask. Go ahead.
So like, how do you get from tertiary to like, the other one, like, yeah, like, I get it, but how do you describe it?
I'm not exactly sure what you're asking.
It goes for like a quiz, for example, how would you describe tertiary compared to the other?
I might say name the structure for a single protein chain. Name a protein structural level of organization for two chains together.
Okay, that's a single chain.
That's one protein chain, that's one string of amino acids. This is two strings of amino, or 4, or it's 6, or 10, all joined together. That's all quaternary structure. Primary is order of amino acids. Secondary is alpha helix and beta sheets. Tertiary is one chain. Quaternary is 2 or more chains. All right? Thank you for asking. Now, before we go any further, let's take a look at fibrous and globular proteins in your notes. Take a wild guess and tell me what shape fibrous proteins might be. Fibers, strands, stringy things. Fibrous proteins are proteins like collagen. Collagen is a great example of a fibrous protein. Fibrous proteins are real good at providing structural frameworks and mechanical support. If you inject enough collagen in your lips, you look like a duck. Thank goodness that fat is going away. You guys remember seeing the internet thing where you just put your mouth inside a bottle and you suck really hard, make your lips really big for a while? Another stupid internet challenge. Anybody do it? I'll do it. I'll put your hands up. But people were damaging themselves. Speaking of stupid internet challenge, you heard of the ice challenge? You put ice and salt up against your skin and hold it there. That makes great third-degree burns. You damage your tissue all the way through your skin. I'm a certified professional. I do my own cryosurgery with dry ice. Probably don't want to try that at home. But it's just like one of the dermatologists, except cheaper. Go ahead.
I was scarred from the dry ice
Oh, really?
on my arm.
Yeah, they'll do that. I mean, it'll burn clean through all your skin down to the tissue underneath and destroy it. And it doesn't heal very well because if you get rid of the skin, you get rid of all layers of the skin, it doesn't grow back. It's just scar tissue, which has a lot of collagen in it. So collagen is a good structural protein. Fibrous proteins also provide movement. Your muscles have proteins called myosin and actin that make your muscles contract. We're going to learn all about myosin and actin about the way through the course. Myosin and actin make your muscles contract. They're fibrous proteins. Take a wild guess and tell me what structure globular proteins might have. Go ahead and say it. Globs. Globs are just balls, things, misshapen, weird things. We also call those functional proteins because they often have pretty specific functions. They do that big giant list of stuff 1 through 6. If you want to be fancy, in your textbook, there is a list of protein functions. On page 49, they turned it from a table into a picture. There's protein functions. We're going to get much deeper into those. Let's watch some more video.
For example, hemoglobin forms a pocket of 14, a small molecule with a higher atom in the center that binds to oxygen. Quaternary structure. 2 or more polypeptide chains can come together to form one functional molecule with several subunits. The 4 subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas of the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The 3-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system.
All right, under your functions for proteins, globular proteins, defense. This is what we're talking about. Proteins, well, let's say, antibodies are a type of proteins. I want you to know what antibodies are. Antibodies are really important. If a foreign invader gets inside your body, your immune system wants to kill it. Make it look scary. Anybody scared of coronavirus yet? Probably okay. It's mostly killing old people. And I'm probably the oldest one in here, so you guys will be alright. But that's a foreign invader, a bad guy. And that has cell surface markers on the outside. Made of what? Carbohydrates and some protein. That say, this is me, this is who I am. In your blood, there are sometimes antibodies. Antibodies look like Y's. They have sticky ends here. So this antibody is going to stick to these things. It's very much shape dependent. If I had another antibody that looked like this, would that fit on those? No. That's an antibody against something else. But we have antibodies that stick to bad guys, stick to foreign invaders. And foreign invaders don't last long with antibodies stuck to them. Hopefully this didn't happen to you, but if you came to the new school like halfway through the school year and didn't have any friends and were walking down the hallway and somebody put a sign on your back that said, "Kick me," you might have got kicked a couple times walking down the hallway. That's like a flag that says, "Kill me." The body's immune system says, you got an antibody stuck to you, we're killing you. So antibodies make up an important part of our defense.
The hormone insulin is a small stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level.
Regulate metabolism. Another function on your list. Insulin is a protein. And insulin helps you regulate your blood sugar level. Insulin helps you regulate your blood sugar level. What does it do? In general terms. Everybody know? It lowers your blood sugar level. Insulin causes your body to burn up sugar and store sugar. It lowers your blood sugar level. Now there's an opposite protein called glucagon that raises your blood sugar level. Insulin makes it go down, glucagon makes it go up. And together these two things maintain homeostasis. They use a negative feedback loop. You got too much sugar, insulin brings it down. You don't have enough sugar, glucagon brings it up. Does that make sense to everybody? Kind of apply some words we use, everybody's okay? Alright, so regulate metabolism. Insulin helps you keep your blood sugar level normal.
Alpha amylase is an enzyme that begins digestion of starches in our saliva.
Okay, enzymes. Enzymes catalyze reactions. Enzymes catalyze reactions. They cause reactions to occur. Anything that ends in ASE, A's, is going to break stuff down. anything that ends in ASE is gonna break molecules down. We'll talk about characteristics of enzymes in a minute. But that's what they do.
The calcium pump is led by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction.
Okay, this is transport. Proteins help transport molecules from one place to another. Proteins help transport molecules from one place to another. Here we're moving calcium. And you're gonna learn all about this sarcoplasmic reticulum thing when I do muscles.
Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form.
You can add storage to your list of functions, because that's not in your notes. In this case, protein is helping you store iron. Iron is not soluble in water. You can't carry straight iron through your blood. It doesn't dissolve. But what do we need iron for? We need a lot of iron. What do we need it for? Make hemoglobin. Yeah, hemoglobin. There's 4 irons in each hemoglobin molecule. So we need to have iron, but we can't just have it sitting around. It'll rot. Well, not exactly, but it doesn't dissolve in water. So protein stores hemoglobin, and protein stores iron for hemoglobin.
Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines.
Alright, so there's our structural framework, mechanical support, and movement, functions of fibers, proteins. One more I want to talk about, actually 2 more I want to talk about. Proteins help regulate pH in your blood because they serve as buffers. Proteins serve as buffers in your blood. 60% of the protein dissolved in your blood right now is albumin. You guys ever hear of albumin before? Might have had some for breakfast. Maybe not today, but over the weekend. What am I talking about? Egg whites. Egg whites are albumin. You got that same goopy stuff dissolved in your blood. There's a bunch of it there, and that helps buffer your blood pH. Molecular chaperones. Proteins also serve as molecular chaperones. Molecular chaperones help other proteins fold properly. Molecular chaperones help other proteins fold properly. Any guys work retail? Banana Republic, Wet Seal, anything like that? They got the little folding things. You make perfect folds in your t-shirts, like boom, 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 all folded, ready to go. You know what I'm talking about? That's like a molecular chaperone. It helps other proteins fold properly and become functional. Because if they don't fold the right way, they don't work. They won't have the right shape. So molecular chaperones make sure everybody is holding the right shape. Everybody okay? Protein denaturation is when you change the shape of a protein. Protein denaturation is when you change the shape of a protein. If you change the shape, it doesn't work anymore. So you do not want to denature the proteins in your body. Let's take albumin for an example. If you heat up albumin, what happens to it? It solidifies. It goes from clear, goopy stuff to white, rubbery stuff. Egg whites. So if you heat your blood up enough, that could happen. Now you're gonna have other issues if you heat your blood up that much. But you can see if you heat something up, you change its shape. If you change its shape, it doesn't work. That's a denatured protein. We use protein denaturation when we cook all the time. We heat stuff up and we change its chemical properties. We can also use acid or base to denature proteins. If the environment is too acidic or too basic, you actually change the shape of the protein. A good example of that is ceviche. How many of you guys know what I'm talking about? Ceviche is awesome. They take raw fish and they put it in with lime juice and it cooks. It doesn't cook with heat. It cooks because it's acidic. You guys, most of you have seen raw fish, right? It's kind of translucent and stuff. When it's cooked, it's more opaque. When you get ceviche, it looks like it's cooked. It has cooked, but with acid, not heat. Both cases denature the protein, changes their shape. How many guys like raw fish? Yeah, yummy. How many guys like sushi, sashimi? Not that California roll crap. I mean real fish. Yeah. Would you rather have totally fresh or previously frozen?
Totally fresh.
No, not the right answer. Pretty much everything that you eat coming from a sushi restaurant or wherever has been frozen before to kill the parasites. I know guys who go out in the Gulf and they catch a tuna and they'll fillet it right there in the boat. You're not supposed to do that, so I'm not going to tell you who it is. But they fillet it right there and they eat it. Yeah, real fresh sushi. Yeah, you're also getting worms. There's a worm called Yankariasis worm. It's about that long. It'll bore through your large intestine and swim around in your peristaltic. It's not something you want. When you freeze it, the worms are dead. So you don't have to worry about it. So flash freeze it, quick and cold, warm it back up again, beautiful. Straight raw out of the fish? Maybe. How many guys work in a seafood restaurant? Get amberjack fillets in? You've seen AJ all shot through a worm. So yeah, especially the big ones. That's like, you might want to cook that before you eat it, because the worm protein blends right in with the fish protein and it's all dead. Put some sauce on it, you'll never know. Let's get one stuck in your teeth. No, totally joking. Hopefully the restaurant you're in doesn't serve that. But raw fish is good, it's been frozen already. Raw, maybe not so much. That's protein denaturation. Look at page 49. There is a very fancy figure. I already told you about that. For functions of protein. Page 50. Picture of that. Page 53 shows you how enzymes work. I'm not going to ask you anything about holoenzymes and haploenzymes. Just not going to ask you. Cofactors and coenzymes. Those are inorganic chemicals that help enzymes do their job. I'm not going to ask you anything about cofactors or coenzymes either. I do want you to know how enzymes work. Page 53 is a great picture of it. Enzymes are shaped to hold molecules close together. And if you hold them close together, you can make them form a bond. So that particular enzyme holds two potatoes close together and sticks the potatoes together so you have a double potato. Well, holds two molecules together and sticks them together so you have a disaccharide. Does that make sense to you guys? That's why shape is so important. If they're not shaped properly, the molecules won't sit next to each other and it doesn't work. You guys get that much, I'll be fine. You want me to see if I can find a real quick video on enzyme actions? Yeah, okay, let's see what we can find. And, 5 minutes is too long, 4 minutes is too long. All right, here we go, minute 54 seconds.
Enzymes are protein molecules that speed up chemical reactions in the cell. A special region of the enzyme, called the active site, is the area where enzyme activity takes place. An enzyme works by binding to molecules, called substrates, at its active site. The job of the enzyme is to convert the substrate into a different product or products through a series of chemical reactions. Following the reactions, the products are released, and the enzyme is free to act on another substrate.
There it's
At
tearing
the
it
active
apart.
site, the substrate should fit into the enzyme, almost like a key fits into a lock. Because of this, most enzymes can fit only one substrate. To accomplish this lock and key fit, the active site undergoes a slight change in shape in order to better accommodate the substrate.
Okay. Substrate, what's the word substrate mean? You hear it all the time, and I've never heard a really good definition that makes sense. Anybody know what substrate is? Write this down. Substrate is the thing an enzyme works on. A substrate is the thing an enzyme works on. Everybody okay?
This is called the induced fit model, because the enzyme is induced to undergo small changes so that the substrate can achieve optimum fit. All enzymes have optimal environmental conditions that are correlated with maximum enzyme activity. Environmental conditions, such as pH and temperature, play a role in how efficient the enzyme is in conducting its reactions. When conditions are less than optimal, an enzyme will lose its configuration and slow its activity.
That's called being denatured, right? If you heat it up or put it in a different pH, look, pH and temperature. If you heat it up or put it in a different pH, it changes shape, it denatures, and it doesn't work anymore.
This is due to a change in the three-dimensional shape of the enzyme and is called denaturation.
Any questions? Shoot.
Are there like forms of denaturation that happen, just like, that they can happen like daily, monthly, yearly?
Usually, when you denature a protein, it's done. Some of it's reversible. If you heat it up a little bit, it might change shape a little bit and then bring it back down to temperature, it might be okay. But if you heat it up past a certain point, it breaks and it's done. Usually. There's a couple odd balls, but.
So there are things that like, if you're running a fever, some proteins.
Some of the proteins involved with a fever actually work a little better. The idea of a fever is it speeds up the reactions or normal reactions in the body a little bit, and it slows down the reactions of the invader. Because those guys, the pathogens, are optimized to run at 37 degrees C at 8.6. That's what they run best at. So your body heats itself up, make them go a little slower and make us go a little faster. Get a good start on it. Does that help at all?
Yeah.
Okay, cool. Over here, yeah.
What happens in the body when like denaturation occurs?
If you denature the proteins in your body, something is wildly wrong and you're in deep trouble. If your pH gets too low or too high, I think I already told you this, what's your normal blood pH?
7.35 and
7.35,
7.45.
7.45. If your pH gets too low or too high, you start denaturing proteins and you die. So it's gotta be in a pretty narrow window. That's why there's buffers and stuff like that. Other questions? All right, now, nucleic acids. Take a look at page 54. And there are nucleic acids. If you look at the top part of the picture there, you see nucleotides. Each one of those is a nucleotide. You put nucleotides together, and you get a nucleic acid. At the bottom of the page, you can see the double helix. Kind of like a spiral staircase, right? And remember I drew it up here and drew the spiral staircase and then turned it into a ladder and zip down the middle? If you look, see the A and the T has like some dots in there, and the C and the G has some dots. Those are the hydrogen bonds that hold it together. Those are the hydrogen bonds that hold it together. And see the purple ribbons on the side? That's phosphate and the sugar, actually called deoxyribose. Sugar called deoxyribose. So that makes the sides of the ladder. And the bases make the rungs of the ladder. Look at that picture up above, the top picture, you see phosphate, deoxyribose, and a base. Everybody see that? The phosphate and the deoxyribose are the rungs of the ladder. And the bases, the phosphate and the deoxyribose are the sides of the ladder. And the rungs are the bases. Does that kind of make sense? Let me draw it. Yes, no, maybe okay. There's the DNA, right? Kind of springy, coily, spiral staircase looking thing, right? You straighten it out. There it is like the ladder. Along the sides, we have sugar, and phosphate. Making up the sides of the ladder. And then here, we have the base. That's the base, that's the base. Those are all the bases. Does that make sense to everybody? That's what the structure looks like. Now the information, the place where the coding is, the coding that determines primary structure is in the bases. If I take this and I split it apart, if I split it down the middle, I end up something looks like this, right? Half of it And I got base, 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 base. Three of these equals one. Amino acid. Three of those bases say, I need this amino acid. And then three more. These guys equals another. And these two guys. And that's how I determine primary structure. DNA is the recipe for protein primary structure. How do you feel about that, okay? That's the structural part I want you to know. Don't worry about the crazy stuff. Oh, by the way, how many of you guys know the significance of that? Those are the nucleotides. So this guy might be a G, that's a C, that's another C, there's a T, there's an A, there's an A, G, C. And that's the code. G always goes with C, A always goes with T. If that's a G, that's a C. If that's a T, that's an A. If that's an A, that's a T. C, that's a G. They go together. Everybody okay? All right. RNA and DNA. RNA and DNA are very closely related to each other. RNA and DNA are very closely related together. The only big, well, there's a couple differences. The one difference is the sugar. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. In DNA, the sugar in the backbone is deoxyribose. In RNA, the sugar in the backbone is ribose. Take a wild guess and tell me what the difference between deoxyribose and ribose is. Close. Deoxyribose has one less oxygen. That's why they call it deoxy. Deoxy. So they're really closely related to each other. I don't care that you really know that. That's more kind of like an ooh, that's cool thing. What I do want you to know is the difference between the function of the two molecules. All right. DNA stays in the nucleus. It's a super secret recipe for Coca-Cola, for KFC, 11 herbs and spices. It stays in there. Doesn't go anywhere. It's preserved. RNA goes out into the cytoplasm of the cell. That's used as the recipe cards to make protein. It's used as the recipe cards to make protein. Now, obviously, the super secret recipe for Coca-Cola is in Atlanta or wherever it is. But they gotta make Coke across the whole nation, across the whole world, so they have to send stuff out. Like the DNA is in Atlanta and the RNA is all the Coke factories out there. That's kind of what I'm aiming at here. DNA is all the information preserved and not used because you don't want to change it. RNA are copies of single proteins that are sent out to be made. Does that make sense to you guys? DNA is the recipe book. RNA is like the instructions for one protein. We'll talk a little bit more about how that works when we get to cells. And also on that I think it's across the page in your book, was it 53? No, 55. 55, there's a picture of ATP. Some of you guys know ATP is what? Adenosine triphosphate, awesome. It is the energy molecule of the cell. ATP, adenosine triphosphate, means there's 3 phosphate groups on it. There are 3 phosphate groups on it. I'm going to greatly simplify the structure of ATP. That's how I draw ATP when I'm in a hurry. The A is the green part and the blue part, the adenine and the ribose. And the P, each one of those P's is a phosphate group. So you see there should be a bunch more oxygens and stuff, but it just makes it busy. That's ATP, adenosine, and 3 phosphate groups. That's energy, that's gasoline for the cell. The cell uses that energy by breaking this bond. It breaks that bond. And then you're left ADP, adenosine diphosphate, inorganic phosphate. Energy, poof. That releasing of the energy allows your body to do stuff like contract muscles, send nerve messages, transport ions across membranes, all kinds of funky stuff. But that's how the body carries energy, how it gets used. And then after you use this, we're pretty good at recycling. After you use this, your whole metabolism, the whole energy creation pathway is all about putting that P back on the ADP so you have more ATP. You break it off, use the energy, you stick it back on again. Break it off, use the energy, stick it back on again. Break it off, use the energy, stick it back on again. Over and over and over again. ATP. How do we feel? Sort of okay? Don't be scared. If you can remember ATP is the energy molecule of the cell, you break the P off, use the energy, stick the P back on, and use it again, you'll be good. For now. Okay? Alright. Done with that chapter. Everybody say, yay.
Yay.
It's over. Now we're going to get into the cool stuff. Tell you what, I want to introduce you to Crash Course as a learning tool. Crash Course Biology, Crash Course Anatomy and Physiology. Monster, really cool learning tool. So let's watch a little bit of this. This happens really fast. You have to watch these a couple times to get everything you say.
Oh, hey, I didn't see you up there. How long have you been waiting in this line? We were like 15 minutes, it was freaking freezing out here. I mean, but demanding you have to feel to do this club. Well, while we're here, I guess it might not be a bad time to continue our discussion about cells, because cells, like nightclubs, have to be
You're not missing anything.
They can all work if they let in the stuff that they need, and they kick out the stuff that they tell me, like trash and ridiculously drunk people and Justin Bieber fans. So no matter what stuff it is, it has to pass through the cell membrane. Some things can pass really easily in the cells without a lot of help, like water, oxygen, but a lot of other things, sugar, nutrients, signaling molecules, steroids, well, they can't get in or it'll take a really long time for them to do it. Yeah, I can relate.
Actually, this isn't the best one. So let me go back, I apologize, and just do this one. I'll show you Crash Course on this, but remember Crash Course.
All cells have an outer membrane, called a plasma membrane, which acts as a boundary between the outside and inside.
You should have your notes out in front of you, because we're gonna talk about stuff.
Some membranes are primarily made of phospholipids, which are large lipid molecules, similar to triglycerides.
Alright, so what's this ball up here, what is that? Phosphate group, what's these two things hanging down? Fatty acid chain. Fatty acid chains. Which end of this is polar? Phosphate group is polar. Fatty acids are non-polar. Which end is hydrophilic? Phosphate. And this is hydrophobic.
Like triglycerides, they contain glycerol and fatty acids. But they differ from triglycerides in that they have a phosphate group rather than a fatty acid attached to the third carbon glycerol. The phosphate head of the molecule is hydrophilic, water-loving. This means that it interacts well with water. The fatty acid tails, however, are hydrophobic, or water-hating, and do not interact well with water. Because of these unique properties, when placed in a liquid environment, phospholipids tend to arrange themselves into a phospholipid bilayer, so that only the hydrophilic heads interact with the watery environment, and the hydrophobic tails crowd inward, away from the water.
That's a phospholipid bilayer. I drew one of those up last class. If you turn that into a ball, that is a cell. Inside has the nucleus and all of the stuff, outside is the outside environment. The phospholipid bilayer, it's phospholipid because it's made of phospholipids, it's a bilayer because there's two layers. Now they call this the fluid mosaic model because it's not solid. Here's something you can do at home. Go home, fill up your bathtub about half full of water, and then put enough ping pong balls on it to cover the whole surface. That's kind of what the outside of a cell looks like. And you can take your hand and go splat. The balls will go, like this one will go like this, but they'll all kind of stay together. That's how I want you to imagine the phospholipid bilayer. It's kind of fluid. And the stuff else hold it together, there has to be, but it's kind of fluid.
The plasma membrane of a cell also contains other molecules that aid in its function. There are a number of different proteins embedded in a membrane, and they serve a variety of functions. Some are involved in the transport of substances across the membrane, while others help in cell-to-cell communication.
Okay, they're getting a little ahead of us now. Let's take a look at membrane lipids in your notes. Are you guys okay with the phospholipid bilayer? Next class, I'm going to draw a big giant cell up on the board. Well, part of a big giant cell up on the board. But you guys okay with the idea of a phospholipid bilayer? Glycolipids. What's glyco mean to you?
Sugar.
Sugar. A glycolipid is one of these with a sugar hat, with a sugar stuck to it, sticking up. Glycolipid is a phospholipid with a carbohydrate stuck to it. If I had to draw it, I'm going to draw it like that. Phospholipid, carbohydrate, stuck to it. Those are found on the outside of the plasma membrane. And those carbohydrates are serving as what? Cell surface markers, yeah. That's where the cell surface markers are. Glycolipids. So I have a fuzzy, sugary coating on the outside of all my cells. Glycolipids. Cholesterol also plays a role in this membrane. Cholesterol kind of wedges in there, fits in between the phospholipids. Cholesterol strengthens and stiffens the plasma membrane. Cholesterol strengthens and stiffens the plasma membrane. So we need cholesterol too. How many guys always thought cholesterol was bad? You know there's good cholesterol too, right? Good cholesterol helps clean all the junk out of our bloodstream. Bad cholesterol puts the junk there. So you want to have more good cholesterol, HDL, than you do bad cholesterol, which is LDL. High density lipoproteins and low density lipoproteins, and we'll learn about those in AMP 2. But cholesterol is found in cell membranes, helps stiffen and stabilize the membrane. Alright, we got a minute left, so let's watch the rest of this just real quick.
Short chains of sugars are attached to the outer surface of some of these proteins, forming glycoproteins, which serve as recognition signals to other cells. Cholesterol is present in the membrane as well. Cholesterol lends support to the membrane and regulates fluidity, giving the membrane the general consistency of olive oil. Phospholipids, proteins, glycoproteins, and cholesterol are all present in the membrane as a flexible pattern, commonly referred to as the fluid mosaic cloud. Together, these components of the cell membrane allow the cell to interact with its external environment.
Alright. Take a look at page 64 and 65 in your book before you come back next class. Page 64 and 65, there's a picture of the plasma membrane and all the stuff involved with it. I understand you might get a quiz on Monday, so don't be thinking, yeah, you don't have to do anything this weekend. Alright, questions, comments, concerns? Lie, be free.